Alright everybody, hello, welcome to my attempts at explaining biochemistry. A little bit late for these videos, just a bit. Had my own exams and stuff to worry about, so have not been very up to date. But we're going to get back on track. Hopefully today I'm going to record like 5 or 6 today, so we'll see how this goes. But yeah, today we're going to be covering nucleotide metabolism. For those of you who are in the class currently, you guys have already had an exam on this. So this is going to be helpful for the final, I guess, if you need to watch this video. And then for future folks who are taking this after the 2026 winter course, this will be great because it should be available to you. So yeah, let's go ahead and get started. So real quick, just want to talk about a couple of things. For the synthesis of nucleotide triphosphates, there's base-specific kinases. So going from the DP form to the diphosphate form to the triphosphate form, we've got, base-specific kinases going from the monophosphate to the diphosphate. So going from monophosphate to diphosphate, there are base-specific kinases. So for example, AMP, GMP, UMP, and CMP, and then these guys go to ADP, GDP, UDP, and CDP. And they always use, these kinases always use ATP as the substrate, because ATP is very abundant compared to the other ones. So yeah, these are specific kinases. So there's adenylate kinase, which uses ATP to make ADP. There's GMP kinase, which uses GMP to make GDP, using ATP as the phosphorylator. And then there's a UMP-CMP kinase, which takes UMP and CMP, uses ATP to turn them into UDP and CDP. After you've turned them into the DP forms, there's another broad kinase called nucleoside diphosphate kinase, NDK. And NDK, what it does is it takes all of the DP forms and turns them into the TP forms. And it always uses ATP. So similar to these base-specific kinases, this one uses ATP all the time. So here's just an example of the pathways here. These are helpful. So like you can see adenylate for ATP, start with AMP, use adenylate kinase, go to ADP, use NDK, go to ATP. See that always the last step here is always NDK for all of them. So yeah, so if you look at, basically it's like that for all of them, right? So you can see here like adenylate kinase, GMP kinase, UMP, CMP kinase. So, so the question they're asking here is why are RNA building blocks so common? And the answer to that is just they're just used everywhere. So like they short-lived, their energy they're used in a lot of places, right? They don't live very long. They have a short half-life, which makes it so they're not permanently in the cell for a while. You know, they help with energy transfer like ATP and stuff, cell signaling, cyclic AMP, and then cofactors like NAD, FAD. Those are all adenosine derivatives and stuff like that. So they're just really, really useful because they don't like stick around for too long, so you can like remake them over and over again when you actually need them. We've already talked about this kind of idea of deoxy, I believe. Like this, so deoxy, all it means is that the 2' OH is gone. So see here we have this is a normal NDP, right, or an RNDP, so a ribonucleotide diphosphate, or we have a DNDP, which is a deoxynucleotide diphosphate. So the 2' OH is removed. And these DNDPs are only used for DNA synthesis and stuff like that. And the main thing to keep in mind about that is that you don't need a lot of DNTPs because you don't really make that much DNA. You during replication, sure, but then everything outside of replication, you don't need it. So like you'll need it during mitosis and meiosis, and that's pretty much it. Specifically during the synthesis phase of those two processes, and then after that, you do not need it. So if you ever get a question asking about like, oh, which one's more abundant, the answer is always going to be the NDP forms. Here is a little thing about how we make these deoxyribonucleotides. There's an enzyme called RNR, ribonucleotide reductase, and what that does is it removes the 2' OH and replaces it with an H. And RNR can only act on the DP version, so it can only act on ADP, GDP, UDP, and CDP, and it turns them into the D forms, if you see here. And then after that, you can use NDK to turn them into the triphosphate forms. Remember, if you just want the normal triphosphate forms, you just wouldn't go through R&R. You would just go straight to the NDK. And here's a little kind of cool, I guess, schematic of what's going on. There's a better one down below, but the main thing to know is that this is a reducing, like you're basically reducing the ribonucleotide, right? So you're going to need a reducing agent, which is going to be NADPH in this case, and the enzyme is RNR. And NADPH is used in this schematic here. I don't think this is very important. It might be something you have to write down on your note card if you really care about it that much, but I've never seen them really test this before that, like, extensively. So, I mean, like, NADPH and this is there, right? So you have to use NADPH to reduce, to oxidize, use the NADPH here as a cofactor to reduce, use it as a substrate to reduce this FADH. So this FAD actually is a cofactor, so make it FADH2, and then this FADH2 reduces the thioredoxin, and then this thioredoxin is ultimately what helps with the process of actually reducing the NDP. And this all occurs in the enzyme, the RNR, by the way, ribonucleotide reductase. So here we've got the ribonucleotide reductase. It's a very complex free radical mechanism. You're not going to have to know like the exact steps, generally speaking. You should know that it's very regulated because you want to make sure you have a balance of the DNTPs. And it requires intramolecular reactions or interactions between two types of subunits, which are the alpha and the beta subunits. So we have the catalytic subunits and then the radical generating ones. So these ones generate the radical, and then the alpha ones actually do the reaction. And so the complex only gets active when you have this alpha 2 beta 2 complex, which is a tetramer. And it's quite heavy. So here's like an image of what we got here. So this is the alpha subunit, or an alpha 2 subunit. So two alpha subunits stuck together. This is catalytic, and then these two here are free radical generating. So there's two different active sites that form between the two alpha subunits and the two beta subunits. Basically what you should see here is like there's an active site like here and here is how I'd probably describe it. So yeah, you can see how like in this image here we have this active site right here and an active site here. So you can do two at the same time. And remember, these beta subunits, these down here are for the radical generation, and then these up here are for the catalytic sites, right? So these are mostly where the catalytics actually takes place. So yeah, here's the mechanism for how this works. I'm not going to go over this, really. I don't think it's that important. I think the main thing to keep in mind here is, like, if you look at this, just track this H along. You can see, like, it's H, then the H is still here, the H is still here, and then now it all of a sudden gets flipped up here. So I would say that's something you can keep in mind. They're not going to ask you, like, individual steps, though. That, I mean, as far as I'm aware, I've never seen them do that, so I highly doubt it's going to happen. But you never know, I guess. So the question is, how does RNR balance the synthesis of the DNDPs? Actually, real quick, I do want to mention something. There are 3 different sites on RNR. There's the active site, the regulatory site, and the allosteric, like, effector sites. So there's like a... Where is it? I'm trying to see. Yeah, like, see, there's like a regulation site, a specificity site, a regulation site, and an active site. So I'll talk about that in a second, but they all do different things. So, like, these two right here, the specificity site and the regulation site, are not the same thing. So, I already talked about this stuff. It's not really that important, I would say, but let's keep going down here. The most important part is yet to come. So, yeah, so this is a really nice slide. Just tells you what binds where. So the RNA regulation at allosteric sites, and its activity and substrate specificity. So with the active site, the activity site, not the active site, the activity site, which is going to be this main primary regulatory site here. There's 3 different sites: the specificity site, the activity site, and the catalytic site, right? That's the names we should always be using when we're discussing these sites on the RNR. So specificity site, activity site, and catalytic site. So in the activity site, only two things can bind, ATP or DATP. And this controls overall is RNR on or off. So ATP activates RNR, and a DATP turns it off. And that should make sense because it's like feedback inhibition. Because RNR makes DATP, and so if there's too much DATP, it's going to shut itself off because it doesn't need to make any more DATP. So that's the activity site. The activity site is RNR on or off? The specificity site is what decides which nucleotides bind into the catalytic site. So here we can bind ATP, DATP, DGTP, or DTTP. Right, so these are all the options that we could bind here. And these are things you just have to memorize, I think, or put on your note card. And what these trigger, right? So these trigger the binding of normal NDP molecules to the catalytic site. And here is the regulation in its full glory. So you can see here that when you have DATP bound, RNR is just turned off, and it's bound in the activity site. So once it's bound in the activity site, nothing happens, it's not useful. However, when ATP binds, right, so let's say ATP binds instead, that turns RNR on. Next, if we bind a DATP molecule in the specificity site, what that results in is it results in CDP and UDP being turned into DCDP and DUDP. Although you'll see that, like, we talked about this before in a previous video, I believe, that to go from CDP to UDP, you actually have to convert UDP to CDP. UDP doesn't really exist for that long. Or DUDP doesn't exist for that long, I should say specifically. And so you can see then that after we make this DUDP, this actually displaces the DATP in the specificity site, and this DUDP is turned into DTTP, right, through thymidylate synthase. So that's where that's coming from. So we make DTTP, and it displaces DATP. And then once DTTP binds to the specificity site, then we bind GDP and turn it into DGDP. And then this DGDP replaces the DTTP, right? And then the GTP, allows ADP to bind to make DADP. And then that gets phosphorylated by NDK, right, nucleotide diphosphate kinase, and binds to the activity site and turns it off. So these all make DP forms, right? So if you look at this, we've got a DP, a DP, another DP. Those DPs can all be phosphorylated by NDK to make the triphosphate form. So we can go from DGDP to DGTP or DADP to DATP pretty quickly. But I would say this is just a slide that you should be studying. You should try and memorize this if you can, because then you don't have to write on your note card. But if you don't want to memorize it, write it on your note card. It is perfectly fine to do so. Here we've just got the structural rearrangements and stuff like that for RNR. That's pretty much all there is to it here. I mean, we have, originally, right, the alpha-2 and the beta-2 dimers have weak affinity for each other. So they don't really bind under normal conditions. But eventually they will associate to form this alpha-2 beta-2 subunit where all of these sites are available. But they only do that when the alpha subunit itself has all three sites bound, right? So that should hopefully make sense. So, like, we don't want this enzyme active if we don't need nucleotides. So what happens is the alpha subunit basically dictates, oh, are we going to turn on RNR or not? So if all three sites are occupied, so if the specificity site, if the activity site has if the activity site has ATP, and then the specificity site has DATP, and then the specific or the catalytic site has CDP, for example. Then once all three of those are bound at once, then these two will connect to each other, and then we'll start doing the reactions. But it won't do the reactions if only two of them are bound. So say that this CDP isn't bound, then it won't do the reaction, right? The same thing goes for the rest of these. So if I bind ATP first, and I bind DTTP, and then I don't bind GDP, it's not going to work. And then similarly, we go the same way here, right? Let's say you need to have all three bound together to actually to get RNR to do its job. And so, yeah, it forms the active alpha beta 2 tetramer, and the reaction goes from there. When DATP gets high, what happens is that it binds to the activity site and promotes a structural change where it kind of like opens up the ring, and that results in an inactive oligomerization. And there's no like way for the free radical to transfer in that, which is why it stops working. Cool little piece of information about it. Not super important, I think. I think it's helpful to know like the tyrosine free radical is generated in the beta subunits, and each beta subunit. And that this is something you just gotta put in your note cards. The propagation of free radical continues amongst the aromatic residues to reach this and is 35 angstroms away. So like basically the tyrosine, just like generates this free radical, and that goes into the cysteines that we've talked about. I kind of mentioned briefly up here a little bit. And so once you do that, or I didn't really briefly mention it, did I? But you get the idea. Basically, the free radical is made. Where did it go? The free radical is made, and it's used to help with this reaction here, and it does that through a cysteine 439, which is in the active site. If I'm not mistaken, I believe it's one of these. I can't remember which one it is. They don't really tell me here. But I mean, like they show you in this, right? So they show you that tyrosine here, on that tyrosine here, and then this cysteine. So the radical goes from here to make this reaction work. And I'm pretty sure that cysteine is supposed to be one of these two, but yeah. You can see when the rearrangement happens, they split way farther apart, so it's no longer 35 angstroms. 35 angstroms is a perfect distance for them to actually jump that radical, but in the alpha 4 beta 4 octamer, it doesn't work anymore. And remember that this octamer is two of these like alpha 2 beta 2 things combining. Right? That's the inactive form. Basically, two of these RNR complexes combine to form the inactive complex. I'm not going to harp on this too much. It's important to maintain DNTP balance. You don't want too many of them. That leads to mutations. The activity site only is modulated by ATP and DATP levels. And DNTP levels are not equal, but they fluctuate pretty frequently. So this is just the pathway for UMP to DTCP. I believe this was. I think this was like not really. I guess it wasn't really talked about in the earlier lectures, but ooh, big yawn. Okay, so. The pathway for UMP to DTTP is a bit of a mess. So let's start with this. So CDP and UDP. So you make CDP and UDP. From there, we make RNR. We use RNR to reduce them to their D versions. Then we kinase them. We turn them into the DCTP form and the DUTP form. Now DUTP does not live for very long. It's very short-lived, and it's rapidly converted into DTTP. So basically all of these reactions down here occur extremely quickly, like ridiculously fast. So say you want to turn some of your cytosine into uridine. All you have to do is deaminate it, right? Because remember that cytosine is just uracil, except it has the amine group on the top carbon, except for has the amine group on the top carbon instead of the carbonyl group that uridine has. So deaminate it, turns it into DUTP. Then you use DUTPase, which phosphorylates it, and then you use thymidylate synthase, which adds a methyl group. And this right here, if I'm not mistaken, uses THF, or tetrahydrofolate, which is I think 510 tetrahydrofolate, which is a cofactor that drops or dumps methyl groups onto their substrates. Once you make DTMP, you use thymidylate kinase to phosphorylate to DTDP, and then NDK to make DTTP. And here is what I was talking about. Thymidylate synthase uses N5N10 methylene tetrahydrofolate. And this methylene group is what is being used as a donated 1-carbon methyl group. Like I said before, after that we just do DTMP. We use thymidylate kinase to make a DP, and then NDK to make a triphosphate. So folate deficiency. If you don't have folate, right, so THF uses folate, then what you actually end up with is incorporation of uracil into your DNA, which causes strand breaks, causes mutagenesis, leads to all sorts of messed up stuff, like a bunch of messed up proteins and all sorts of stuff like that. Folate deficiency, I believe, if I'm not mistaken, in pregnancy, right, a lot of women are told to take like folic acid supplements or folate supplements. And the reason for that is because, believe it or not, a baby or an embryo, right, is a cell. It starts as a single cell, right, that is constantly dividing. And so you need to make sure that you're providing the baby with ample supplies of folate, because it's constantly dividing over and over again until it reaches a stage where it's actually, you know, like a full-fledged human being. So before then, though, you have to make sure that the mother has all the folate that she needs so that she can actually, that folate can be used by the baby, right? So the baby can use that folate as a cofactor to make sure that it can make nucleotides properly and DNTPs properly so that it can keep replicating over and over again. So that is why folate is very important, and why it's probably been prescribed to, maybe some of your parents actually had folate prescribed to them, or some of your moms had folate prescribed to them while they were pregnant with you, or a sibling or something like that. I know my mom was, so. Here is a diagram of all the nucleotides in this stuff. Not gonna go over it in depth. You guys, I already have videos on it, and you have your lecture videos here from your professor. We talked about it pretty well up here, anyways. So, yeah. But other than that, actually, that's actually the end of this video. I hope it was helpful. If you have any questions, feel free to come to the IC and ask. But other than that, happy studying.